everyone. Thank you so much for coming to today's talk. It is my pleasure to introduce Dr. Scott Yanco. He studies individual movement across their individual's life cycle and the multiple dimensions of their niche as they move. This to inform population level dynamics. And that's a very difficult analytical problem. But Scott does this by creatively incorporating different approaches. And I think that's why his research is a big contribution to the advancement of movement ecology. He did his PhD at the University of Colorado, Denver, then a postdoc at Yale and one at University of Michigan. This year, he was also selected as one of the elected members of the American Ornithological Society. And he recently started his position at the Smithsonian Migratory Bird Center, which is nearby. So hopefully we'll have more chances to interact with him. So welcome him to Valentina,
thank you so much for the invite to come here. It's been great already this morning meeting with and getting to hear more about all the great work going on here. I'm looking forward to more conversations this afternoon. And yeah, for I was up to DC, so it's like to get to know the extended neighborhood as I dive into this position. And I'm excited to tell you a little bit about my research here today, which, as Valentina told you, is really trying to build from individuals to higher level ecological. So think about these individual scale mechanisms and leverage really a burgeoning technological revolution to try to understand and rethink classic ecological theories and conservation approaches. And for me, a lot of it comes from thinking about landscapes like this. This is the Champlain Valley in northern Vermont. It's one of my favorite landscapes, but you could picture a landscape like this really anywhere. And I love this picture because you see all this heterogeneity in space. And you see signals of the heterogeneity. Obviously, there's the different habitats, there's aquatic and terrestrial environments, there's elevational bands. You see the leaves changing, right? Seasons are literally happening in this photo. And you see the impact of people, see agriculture, development, roads. If you look closely, you can see people on that landscape. You can see cars driving down the roads. The dynamic elements of human disturbance. These are all things that animals have to cope with. We have a variety of ways to cope with these, and all these adaptations and responses to these really complicated environments ought to scale up to produce effects at population levels and beyond, inform our understanding of even macroecological processes. So that's what a lot of my research focuses on. So I'm going to talk to you about three broad themes today, building from here. And the first one is thinking about how this nexus of these complicated dynamic environments influences animal behavior and how that interaction itself shapes life. We do it by studying seasonal migration in birds, but there are, of course, other ways that you could think about those interactions. The second part will be talking about how we take those life history and behavioral responses to these environments and think about scaling them up to population level processes, specifically individual-based approaches to understand what makes populations go up and down, what makes the villages change. And then the last little bit of the talk will be sort of forward-looking and thinking about how we can leverage approaches like this, the technology that we've developed, the platforms that we've developed, and what we expect to be able to develop in the coming years to really catalyze a new way of thinking about both new and old questions in ecology. We'll dive into this. The first one first, right? It's a good place to start. So the central question guiding this research is really thinking about how migration modifies organism-environment interactions. We know that these migratory birds, as they transit the Earth, are going across different seasonal regimes. You know, imagine this cartoon here. You have a migrant going from a super high latitude, where the amplitude of seasonality is quite high, down to a tropical environment, low. And it ends up creating this environmental resource curve out of these places, right? So the seasonal environment that this bird experiences is actually an amalgamation of places on Earth. It doesn't actually, we think about it as flattening seasonal resources. So it's sort of the dominant paradigm here. I'll give you a quick example from my own research where we showed this. There's others, and it's not actually even totally the point of this research, but I think it illustrates it well. This is a flammulated owl. This is a species I love dearly. I've studied it for a couple decades now. They're high elevation or mid elevation montane species in the Western US and Mexico. They eat moths. So it's an insectivorous raptor, and they're a partial migrant. So there's a resident population down in the Central Transvolcanic Belt in places like Estado de Mexico and Michoacan, and there's a migratory population in the West. We tracked a bunch of them from a bunch of different sites across the Western United States. We get these tracks from, I think it's 16 individuals, and we can annotate those to environmental conditions. We get all this great satellite data that tells us things about what the temperature was that that bird experienced, what NDVI is, and NDVI is telling us something about ecosystem. And we plot those over time. The gray plots here, the gray points on the left, that are real owls, right? So this is nighttime temperature, the days of the year, and these are real owls. These colored points are showing you what would have happened if they stayed in Colorado, right? And it's sort of obvious. It got cold in the winter, right? And then same thing here, but this is now NDVI. And you can see they end up flattening their NDVI a little bit. These are sort of pairwise individual comparisons. You can see in the winter, they experienced greater ecosystem productivity by going to Mexico instead of staying. Right? So we did more with this paper than that. I promise this was about thinking about where is migration leading or more. And this is how moving ecology works for a lot of these resource tracking, flattening your resource. But you can see things in here that are a little bit distant. During these migratory periods, so the green and the purple here are when the birds are migrating, so not on a stationary ground. You can see the variance in NDVI. It just goes crazy, right? It's dropping down as they're crossing deserts, they're crossing these really arid environments. If you test, it's not shown here, but if you test the variance, there's like a real heteroscedasticity problem here. The variance explodes during parts of the year. It's too temperature as well. It explodes the shoulder of these seasons. You can tell that there's more environmental dynamics here than more by just sort of thinking about, did it get better? Did the seasonal dynamics wow? So, so I want to turn to another project where we tried to increase that complexity. And we did this with not owls now, but cranes, and 4 species of cranes. It's part of a project led by the Max Planck Institute of Animal Behavior over in Radolfzell, Germany. It was a really ambitiously named project. I'll tell you at the end, there weren't 1,000 cranes, there's like 400. Don't get 1,000 tagged down. I think a couple of large global events got in the way. But the idea here was to think about how can we take the tracks from these 4 species of cranes that all have different natural histories, but also similar natural histories, and treat these environmental dynamics as multidimensional instead of just resource time. So we borrow from niche ecology, classic niche ecology that's considered n-dimensional hypervolumes, but integrate the time dynamism that move need. So we do this with a new framework. So all of these cranes, we annotate to their environments so that we know what they're experiencing on a day-to-day basis. We do it with 4 environmental variables. EVI, which is just like NDVI, it's telling you sort of how productive the environment is literally a measure of the Earth is. Temperature has obvious importance. All these species prefer foraging on crops, so crops are really important. So we use the proportion of crops in the vicinity of the GPS point, and they all like roosting in shallow water as a predation way to go to crops, they to forage, they go root in the water, and then we did a thing that we called time-ordered principal components analysis, which is a really fancy way of saying principal components analysis, but you keep your data set in order of time, right? I mean, that's all there is to it. You keep track. So we take individual average conditions for a week, and then we take the average for population for that week, and we put it into this PCA for all 4 of those species. By preserving that time order, you get to do a couple of new things with this PCA that we thought were pretty cool. The first one is ask if there are trade-offs among the niche components, which is something that's not typically thought about. So it might be that when you go to access one resource, you're actually giving up another. We know this to be true from case studies, right? The classic example would be humpback whales that leave really productive waters to raise their calves in really nutrient-poor waters that are predator-free and are more thermoregulatorily advantageous. And so if we see in these loadings, orthogonality, then you know that they're independent. The variance is independent. There's no trade-off. If you see them in opposing directions, then you know that there's evidence that those niche axes are trading off. You can also take the PC rotations and put them back into PC space, just same way you would do to cluster this stuff. But you can you can map the seasons back with the individual niche curve species. In other words, they use the same piece of environmental space as in these different seasons, and how are those related to one another? What are the axes of variation over the year? And then my favorite part is using the PC scores, which we all do. PC score telling you how much each sample contributed to variation on that axis. If you line those up in time, it's telling you about the specific weeks of the year that mattered for that potential for that particular axis. So you can look for patterns related to big life history events or big annual events. Here's just a cartoon example where we just looked at, spring or fall migration versus breeding and wintering time. You could have no rhythm, you could have life history based rhythms, you could still recover seasonality even though they're tracking, right? So I'll show you quickly a few of these results. The first is that there are almost no trade-offs, right? By moving broadly across the year, we see very little evidence of trade-offs among these values. This part is sort of flattening out the time. So the driving hypothesis here, right, is that they're actually using their dynamism within their individualized niches over the course of the year to resolve. We do see one over here in the black-necked crane where proportion to water trades off with EVI, right, over the course of the year. So safe habitat is unproductive habitat. This is also, so again, like this is, you can think of this as the tracks, the GPS tracks of the cranes, the average GPS track of a crane, but rotated into environmental space, so the geographic space. There's very little niche conservancy. The wintering areas had very little resemblance in environmental space to the breeding areas, which is contrary to prevailing theory about what we think migrants are doing. We think that they're sort of like broadly tracking their environmental niche up and down. They're just following favorable conditions. That's not stuff. And then we saw these scores was, again, my favorite part, where we see that exactly that. We still recovered this broad season of seasonality. So again, green here is spring migration, orange is fall migration, and you still see this broad seasonal on the first axis, on PC1. We can even ask what is driving that by looking at what loaded onto PC1. So here, proximity to water and proximity to crop. But notice the EVI and temperature were flattened, right? So now we've moved to this model where instead of are you niche tracking or not, I'm flattening your exposure or not, yes, you're both. They're holding temperature and EVI constant, but they're doing it at the expense of that origin conditions. All four species showed a broad seasonal too in this study. If we go to PC2, which is the second largest axis of variation, you see this really cool spike right on the periods of migration. Right? You say, okay, so the second most important source of variation here, it has to do with environmental conditions while they're migrating. And it's all temperature for this species. And that's because they cross the Tibetan Plateau. That is amazing circular integration. They have to do with these extreme temperatures that are over 20,000 feet in the air. And we can recover that here. And that's like a huge source of environmental variation. Okay, we'll do one more. Concrete, again, here, they're still seeing that big seasonal arc. In this case, what they're tracking and what they're not has flipped. So they're tracking proximity to water and proportion of crops, but they're letting temperature in the area. Here, we get a secondary axis that doesn't have to do with migration, but you get this spike at the end of the breeding season. So I don't know what it is exactly. It's probably chick rearing, could be a molt, could be preparation for migration, but it's driven by proportion of crops. So they're after the chicks reach independence, they're moving much, much closer to crops prior to their migration, right? Which is going to be cut away. Okay, so I've hopefully convinced you that animals are using these complex seasonal dynamics and that they're tagging them with their life history. Not just broad seasonal tracking, so not just following a niche around. There's like a very specific thing that's connected to important events in an animal's life. So that would suggest that that has some impact on an animal's days. I'm going to show, I'll admit in advance, like in a dissatisfyingly superficial way, 3 results to try to that these complicated dynamics of migration are shifting species life history strategies, and that they're changing these individual species roots to fitness, these roots to performance. The first is the pair studies using dynamic energy budget theory, which is a really amazing framework for thinking about metabolic ecology. It has a lot of nice properties that go beyond the metabolic theory of ecology, namely that body size emerges from it instead of relying on erroneous scaling. It's first principles. I mean, it's basically based on like Newton's laws and stoichiometry. You get energy in and it has to go places. It's incredibly mathematically dense, notationally dense. It's written in these really difficult to read papers. Coming to this, I'm a quantitative ecologist, but I came to it from the field, and it's like really stretches my ability. But you wrap your head around it, what you get is energy in, and then this cap-up parameter here is dividing these energy streams into self and into reproduction. To be like ringing your Stephen Stearns bells. This is the trade-off between self and reproduction that we think life history is all about. And you get all these other cool things in here about how much you're devoting towards growth versus fecundity versus operating maintenance. You can think about developmental times for the young. And so Allie Pierce, a grad school colleague of mine, and I published a pair of papers where we looked at dynamic energy budgets, first in seasonal environments, and extended them to match them to an agent-based model in a theoretical context to think about how these life sector parameters might shift in a seasonally fluctuating environment and a seasonally fluctuating environment among which animals can move. So, like I said, dissatisfyingly brief on this slide to establish this point. This first one is just a theoretical simulation. All you need to know is that this is life history, slow life history, time versus itself, fast life history, right? And this is aseasonal, moderately seasonal, highly seasonal. And we have this triangular shape where all life histories are possible if there's no seasonal fluctuation, but you can constantly in highly seasonal, which is between two. You can figure out the body size you have to be fast. After we built that model, we then tapped this agent-based model. Oh, and this broadly that validates macroecologically. When you look in trait databases, you can find evidence to support this being the case. It's not just variance in mean traits, but the set of traits that are possible. We then hitched this to an agent-based model of migration and inequality. Way more complicated. It's hard to tell the story of what we see here. But we do see that migration pushes you into different regions of life history space, which I think is an important part for today. So here's our non-migrants. Here's our migrants. These two strategies over here have to do with like optimizing energy versus just doing the best you can, how to move, what latitudes you're moving to and from. But we see that there's a holistic different life history for non-migrants than there is for migrants, but it's less straightforward than just varying investment itself. So PC2 is what Kappa loads onto here, but there's all this other, this is like how fast you can reach sexual maturation. Factors are really, really important for the differences between the strategies, and there's overlap among the axes, right? If you just look on PC1, these points are kind of in a line. PC2, these ones are right. So it becomes a really complicated thing. The other thing to point out, we know that migration is this incredibly laid out, right? It's on birds all the time, right? Valentina is obviously very familiar with this. This might help us understand some of that. There's a lot of ways to be a migrant. There are more than one life historical configuration associated with migration, probably because of this, because you're dampening seasonal amplitudes and opening up more life history space for yourself. The second approach is also using a physiological model in a partially migratory system. So this was work I collaborated with Nils Linnick, who's at Max Planck. He studies these partially migratory blackbirds. And here it's a partially migratory system where they share breeding grounds. And some of them go south for the Iberian Peninsula or North Africa. Some of them stay, and it's plastic. Switch. So you don't know year to year who's going to go, and you can't tell morphologically. You can hold them in the hand the day before they go. And it's a pretty short migration. They do it kind of slow, so they don't engage with kind of hyperengaging. Can't stop. And so he was really interested in trying to understand the physiology of it. And so what he did is worked with a company to develop internal body temperature and heart rate model that you can put in these birds. And then you pair that with automated telemetry, so we know where they're going, and some previous work, so we can map out their routes. We know who left, who went. We know where the population migrates to. Now we know what their internal body temperature is, and we know what their heart rates are. And the first thing to point out, so there's a lot in this paper that Nils got into about the dynamics. It turns out they modulate their heart rate a month before they go, but we can't tell, right? They're saving up energy. If you were to, this is heart rate. If you were to count up the number of beats over the course of the year, the dynamics are different. but they eat the same number of times. Right? So the total energetic throughput of these birds is identical, assuming identical morphology, which seems to be the case. So that's strange. And then this is body temperature. And you can see the birds that stayed and are in blue, and they did maintain a little bit lower set point by about 0.5 degree. But it was about 5-6 degrees Celsius warmer than the other. So the body temperature difference was not of the magnitude that we expected. And so my part of the study was to actually do the thermoregulatory model here. So we run this through like really classic, well-studied thermoregulatory models from Potter and the niche mapper package, which has extended those and made them really tractable in R. And given the known ambient temperature and the known body temperatures, we can say what their energy expenditure should have been. And then given that we know that their top line field metabolic rates were the same over time, we can measure the size of that energetic deficit. And so here we can see like the migrants save a huge amount, like 18,000 kilojoules by migrating. So even though they're paying the cost, right, this doesn't incorporate the cost of migration. So some of this pays for flying, but it's not that big of a flight. It is definitely, we don't know what the exact kilojoules are. It doesn't cost them 18,000 kilojoules to go from Germany to today. And so we don't know what this does, right? It could be saving up so that you're in better physiological conditions, so that if you get capricious weather events, it's like a bet hedging strategy. I kind of think that it's capital investment in greater quantity, but that would be at odds with this item over here, right? But maybe not. Maybe it's just one of these strategies over here, and we're, there's more work to be done here. But the point is that even in this population, where individuals are switching their strategies year to year, it is pushing them into this completely different energy regulatory regime. It's completely rearranging their physiological strategies just from the behavior alone, again, within individuals. Okay. Hopefully it'll become apparent why I sort of tried to go through that quite fast. The point here is that I hopefully convinced you that migration is modifying ecological niches. It's doing it in really complicated, really dynamic ways, right? It is not just a static species level niche that things are following around. And if we just did enough SDMs, we would know what to do to these things. And that the movements, migration here, but it could be any other movements, the way these animals' behaviors are engaging with those is altering their metabolic organization, which is another way of saying it's altering their life history strategy, altering their orientation towards performance and in fitness. Okay. I started the talk though by talking about all the humans we could see on the environment. And so now just introducing anthropogenic disturbances to this paradigm as we continue moving forward and pushing towards populations. Human activities are likely to modify these and disrupt these relationships. We know that that's species. And because these environment relationships are connected to life history, we would expect that to have an effect on demographic performance downstream, right? We should be able to see this at the population level. And so that's the second part here, where we're going to talk about, think about the mechanisms that underpin population changes. So not just whether humans are associated with them, but what are the pathways from disturbance all the way up to these population level outcomes. So what do we know about how human activities affect animals? We know like it's day to day, right? In a broad sense, it's these things that I might be more to. We have shockingly little understanding of how these things are impacting. There are well-studied systems, to be sure, where we can connect parts of these stories together. But for many of these, we don't know the answer. I was talking with Jeff earlier, and there's this 3 billion birds paper, right? And he's involved in road to recovery. 3 billion birds went missing from North America. This is huge species. Most of them, your guess is as good as mine. We can't tell you what conservation action you should do tomorrow to save trees. We don't really know why they're. I'm going to give you an example of two such mechanisms that I had a chance to study. And then landscape modification and human mobility, right? These we know are big ones. This is habitat loss and habitat fragmentation and disruption and our activities out on the environment. And the reason that we lack mechanistic understanding of these two specifically, but I think it's emblematic of many of them, is that they're typically confounded, right? The picture you see in the background here is Shibuya Square or Shibuya Crossing in Tokyo. And you can see all the stuff is where all the people are, right? And that's a real challenge from just a basic study design standpoint. Under super tragic circumstances, the COVID-19 pandemic actually offered an opportunity to break that confound. So we certainly wish that we hadn't been given this opportunity, but it was a tremendous cessation, disruption in human activities that in many places broke it. So you probably saw during the pandemic stories of animals inhabiting urban environments. Many of them were right, like there were no dolphins in Venice, but some of them were real. There were sea lions in parking lots in Santa Cruz, California, and there were wild dogs on the streets of Kigali. And so a group of researchers put out this paper 5 years ago now, welcoming us to the anthropause, this disruption, and suggesting that this was a chance for us to study some of these effects as like a grand and tragic natural experiment. Thus started the COVID-19 biologging initiative. Brett Jesner's here. He was a part of that as well. I sit on the steering committee of it, and we're really proud of our sort of collaboration model. I'd love to talk to you all about how we did this. We think that we have a really unique collaboration model, but I won't bore you with it today. I do want to show you the results of one of the sub-projects. This is my, I'm not just the president, I'm also a customer slide, right? I don't just sit on the steering committee, but I'm running and stuff. This is work, again, Brett's been involved in this. I'm co-leading this with Ruth Oliver, an assistant professor at the University of California, Santa Barbara, trying to understand how animal movements are disrupted by both human mobility and human infrastructure, and how those two effects interact with one another. We do this in North America or in US only because we need human mobility data. So we need to know where people are moving, and you can only do that in the United States. There are these tremendous and frightening data sets that are collected anytime you have a free app on your phone. You go to place. And SafeGraph typically sells their data for lots and lots of money. They made 2019 and 2020 data available to researchers studying the effects of the pandemic. So we were granted access to it. It gives us daily device counts. So it's not quite as scary. I can't look you up and see where it's totally de-anonymized. I see at a sense of block group level, which is an annoying station. Basically a neighborhood. I can see how many people were out and about on the environment at either like an hourly or a daily time scale. So we take these movement tracks. We have this massive data sets, 37 species. I think after cleaning, it's almost 10 million GPS fixes, 3,000 or 4,000 animals. We have their movement trajectories. We annotate them all to the environment, including these device counts and satellite-derived measure of landscape modification. And then we characterize weekly space use. It's kind of like home range, but not quite, but weekly geographic space use and knit prep, essentially, on the median scales. And then we ask how these things interact. And we see massive effects. So of the species we studied, 91% of mammals and 50% of birds responded in some way to a human activity. That bird number, a lot of our birds are waterfowl that are migrating during this. So it's actually even sort of 50% is high, because I would have actually predicted many of the waterfowl would just kind of fly right over us. You consider the timing of it. And the effects were combined. In other words, for most species that responded, you needed to understand landscape modification and human presence to directly model the effect of humans. In many of these cases, it was interactive. So the human context dictated their response, and it happened in both directions. So it's totally species specific, which highlights that taking just satellite-derived measures of human infrastructure, impervious surfaces, and stuff is insufficient to understand these animals' response. The same was true for niche size. So again, just to walk you through, this bar is our area size aggregated results. This is our niche size results. This is if they only responded to humans, both, or only landscape, or both, and then that is if they didn't respond at all. So of the ones that responded to niche size, mostly you needed to understand both anthropogenic drivers to get this right. And then we were like, oh great, we're going to look for typologies. We're going to try to figure out what the pathways of response are. Do you respond by shifting your area? Do you respond by shifting niche breadth? Do you do both? And it turns out like it's super variable. Some of them, very few only shifted their niche. But for human mobility, it was sort of equal. Did you shift your area? Or did you shift both? Landscape modification is kind of all of the above. It's super context dependent, right? I mean, it's the thing that you keep coming back to over and over and over again in ecology. And the species are different, individuals are different. Context matters. The individual natural history matters. There was no big, broad, sweeping generalization other than birds were a little different than mammals. So this is just to put all the effect sizes of all these species-specific models on a map. So what we do for this is basically take like a scenario of low human mobility and low human modification and a scenario of high, and ask the difference in the predicted responses from our fitted model to try to assess effect size from these big Bayesian and interactive models are a little bit hard to quantify. So that prediction, that difference gives us a sense of magnitude. So if we went from a low human scenario to a high human scenario, what did that change about your response variable? And the effects are massive, right? So 12% contraction on average for mammals, almost 50% expansion in weekly area size for birds. And that's huge, right? These are common ravens. So these are like the Yellowstone ravens, the John Marsliff and Matthias Loretto ravens, 24 square kilometers a week, going from the 20th percentile to the 80th percentile of human modification within that species too, right? So this is not like predicting out of sample within there. Same thing. Coyotes shrunk their weekly species by 18 square kilometers. Next was a little hard to characterize, right? It's like unitless reproduction for environmental variables. But we're seeing single and double digit percent changes in niche breadth. And that's after controlling for species, right? So this isn't just that you shrunk your area, so you experienced a smaller niche. So big impacts. So what does that mean when we think about the landscapes that we're increasingly creating? This is a picture of, this is New Haven, Connecticut, a place I was fortunate to live for a few years. And I love this picture because I think it's emblematic of the places that conservation really needs to happen. 30 by 30, this endeavor to cover 30% of the United States or 30% of the globe by 2030, it's fine, it's great. It may or may not happen. It cannot be the case that means we concede the other 70%. And that's both for the species and for the people. It won't protect the species. And I think that many of us would be happy at protecting the species for their own sake. That's how we got into this work. But I think that our charge is actually to protect the species, to protect human interests. I don't think it's an accident that the United Nations Sustainable Development Goals include life below water and life on land. This is not the Convention on Biological Diversity. Right? This is like how you have a healthy human society, is a healthy biosphere. And that means taking these current extinction rates, which are 1,000 to 10,000 times above background rate, and slowing them down, and doing so in environments where you have mixtures of populations, you have suburbs, you have urban areas, you have this matrix, this complex dynamics of humans moving forward. So, right, if our charge is how do human activities contribute to biodiversity loss, based on the findings that I've showed you so far, hopefully I've convinced you that we need to actually think harder about the components of human activities. Which parts of what we're doing are having which effects? And that we need to do this on a species by species basis. And we didn't see a uniform outcome across all species that's going to give us a silver bullet. We need to think about this species by species. So population biology 101. How do populations change? Well, the combination of the four vital rates, right? It's the BIDE, it's births, plus your migrations, minus your deaths and your emigration. That's what makes populations go up and down. So if we want to reverse that extinction rate, what we want to do is understand which of these and how are these changes of these disrupted environmental interactions leading to changes in these specific biomes, right? That's how we're going to target this type of conservation. Most of our biodiversity monitoring data looks like this. It's unmarked animals. It's observational records submitted via iNaturalist, via point counts, via eBird, stashed into GBIF that we do the Living Planet Index with. We do these big abundance. They're great at telling us that species are declining, but they cannot attribute to any single vital rate. They just can't. It's just not how they work. Right? We don't know the identities of the individuals. So if we want to get to these population vital rates, we need marked animals. And I'm going to make the case to you that this animal tracking that we're just doing this behavioral research with is actually a key to changing the way that we think about these problems. So traditionally, we've partially separated out the vital rates by looking at marked animals, bird banding, ear tags, right? We know the identity of an animal and we observe them at regularized intervals. And you can kind of start to separate things, right? Now I can tell if an animal left my study area or died, right? And I sort of accept that confound and I reason away about why the number of animals that left my study area is probably not that big, or I know what the percentage is, and I accept that directional bias. But these are hard to scale up. They take a lot of time. The detection probabilities are quite low, and you're often restricted to small geographic study areas. Tracked animals, on the other hand, first look a lot like marked animal data as repeated observations of individual animals. A priori, we should expect that we can use these methods, but we also know that we have several key advantages. The observations are not restricted to any single study area in many cases. We can start to separate these emigration processes from the survival processes themselves, right? So now we're actually getting to really precise measurements of vital rates in our best quality data anyways. And we can use all these cool behavioral techniques that I've shown you, and that there's a whole literature in movement ecology. You can look when an animal dies. Best quality data is a GPS with a mortality switch. You have a signal, you get an email in your inbox when your animal died. And then you have to trace what happened before that. Sometimes hourly resolution. All these environmental and anthropogenic variables you can associate with it to try to attribute the causes of those mortalities. So the when, where, why, and how, right? And start to think about proximate versus ultimate causalities, right? Are road collisions really just random events? Maybe certain individuals are pushed into habitats because of their internal state, because of environmental degradation. We don't have a framework for thinking about that currently because we don't observe death very well. Coming from the migratory bird world, anytime we look really hard for it, we see these lagging indicators, these carryover effects where conditions in one season drive vital rates in another season. But we often just don't look for it because we can't. Well, tracking data should help us solve that kind of thing. We can look for these dynamics, right? Where maybe these effects are chronic instead of just acute and associated with the time of mortality. Maybe it's just a very particular time in the innings of the crisis. I'll give you one example of this. This is using automated radio telemetry data with the Kirtland's warblers. We also did this with banding data with black-throated blues. I'm not going to talk to you about this today. This was work led by Nathan Cooper, my colleague at the Migratory Bird Center. And Nate studies Kirtland warblers. He's the world's leading expert on Kirtland's warblers. They wintered down here in the Bahamas, and they all go to this tiny little breeding ground right here in northern Michigan. It's a highly management-dependent species. They're reliant on a very narrow age range of jack pine habitat that has to be 5 to 15 years old. They were just taken off the endangered species list. And we wanted to look for a carryover effect of environmental conditions during the migratory period and the subsequent breeding period, and to get a direct estimate of survival during migration. You don't have that for most birds, let alone birds of this size, 17 grams. It's tiny. And lo and behold, when we use this tracking data, we find, right? So what we did is we take their movements in the Bahamas, we associate it with their environmental vegetation or the enhanced vegetation index, which is telling you dry versus wet. And we have to sort of relax a bunch of model assumptions of the CJS and extend it in ways to work with this messy automated telemetry data. But what we see is, one, that migration survival is lower than the other 2 periods of the year. And we see subtle but significant and important effect of winter vegetation conditions on survival during the winter, during their migration to Michigan, and on the breeding grounds in Michigan. Right? So this is a direct measurement of a carryover effect in a 17-gram warbler. You can then take these survival estimates and put them into a simple population model, just like a classic Leslie matrix, and walk that submodel across EVI to ask what happens if the Caribbean dries out, which is what climate change projections tell us is going to happen. And we end up in population growth rate starts to slide down and then absolutely follow up. No amount of jack pine management in Michigan solves this. No amount of monitoring in Michigan sees this. You can only do this with longitudinal data of individual animals to get the actual causes of mortality. The species just came off the endangered species list. And in the most recent census, their populations declined by about 30%. Right? We don't know if this is the mechanism driving that particular decline yet, but it might be, and it should be terrifying to those of us engaged in conservation biology that these kind of effects are out there, and none of our monitoring, very few of our monitoring mechanisms can see them. Okay, so hopefully I've convinced you that human activities are changing these environmental interactions, and that the responses to them might be at least a component of the engine of global biodiversity loss. Maybe I've inspired you with some hope that these individual-based techniques can give us a new way of studying them to both understand basic biological and ecological processes, but also a new way to think about the information we need to do conservation. In the last couple minutes, I want to turn away from the doom and gloom of all the things we are not seeing by not tracking our animals with sufficient technology to talk about what we're doing to try to actually solve that gap, to build these out into larger priorities. To do that, I want to highlight the rapid advances in technology. This is a field stork. This is probably the first tracked animal that we are aware of from Western science. This is a white stork, it's the national bird of Germany. This is back when they sort of assumed in the 1820s that they were migratory, had no idea where they went. And then in the 1820s, a few of these birds showed up. I think it's like in the 20s of them, alive with arrows or spears through their necks. They had survived an attempt at hunting on their wintering grounds. And then ornithologists caught the bird, take the spear out and go to the anthropology department and ask, like, where in Africa would this have come from? And voila, you now have 2 points on the map. You know where those birds went, right? Fast forward, a couple years ago, the folks at Max Planck used a radio tracked a moth. They followed it in an ultralight aircraft. They sought course correcting for wind conditions so that it could navigate through known direction. This is Nils with his internal body temperature, heart rate log on a 100-gram European blackbird. This is me gleefully holding the ill-fated Icarus tag. This is the new 5-gram tag that transmits live feed into your computer from the International Space Station. And then this is nothing totally special. This is one of our automated radio telemetry tags, but I wanted to highlight these wires here. Like, this is pairing movement data, which is also survival data, with field-based experimental methods, right? We're warming up nests and trying to understand it. Work okay. But the point is, all these technologies in a pretty short period of time have completely changed what we can see about the world as ecologists. And the volumes of data are staggering. This is one repository alone, MoveBank. It's the biggest one, but this is already out of date. There are 6 billion observations, of animals in there. Each one of those is a repeated observation of an individual animal that we could do vital rate work with. And it also encodes behavior, and it encodes physiology, it encodes their environmental experience, it encodes biotic interactions. There's just one, but many. You map that onto the globe and you can see things like this. I just love these animations that the Max Planck people. You see these animals moving with the pulse of the seasons. You can see their small scale movements. You can see them moving together. You can see them moving apart. You can see them transiting just inconceivable distances, right? So how do we unlock all that potential? We need to keep pushing on the technology. We need new tools, especially computational tools. Think about the first study I showed you, that was 16 owls. There were a lot of things that I can do by hand with 16 owls that I can't do with 16,000 individuals, right? So that takes new techniques for just basic data processing, data cleaning, data aggregation, and harmonizing. It also takes new communities of practice to make sure that we are overcoming the massive sample biases and where we put tags out. So making sure these are expensive technologies, takes a lot of training. There's a tremendous amount of global inequity in who participates in this research, and it's not an accident. We also need new communities of practice, new people thinking about how we use these technologies and what kind of science we can do with them to get at these new and maybe tackle some old questions. Ruth Oliver and I are sort of calling some of this idea the new ecology of death, which makes me sound like a much darker gothic person than I am, I promise. But the basic idea is that we don't have to get afraid of work. How do we expect and ultimate causes to interact with one another? How would we even think about, okay, let's say we solve the causality, and now we have a good way of understanding the mechanisms that produce individual heterogeneity. What does it mean for macroecology? biology? Like, what if we confront classic life history with this individual heterogeneity? Do our old theories hold? Are there theories that we haven't, that predict individual heterogeneity as important that we haven't tested? And I think, you know, we think quite strongly yes. Part of that is we're launching a new project under the COVID-19 initiative. We're going to take the same sort of approach, disentangling these two drivers, but try to do it with mortality now. And so we've started that. We're building a global data set. We've paired up with Euromammals. So Fran Cagnacci at the Foundation is helping us. If you got data, you can email me or Ruth. Maybe email Ruth because it's, you know, something's about to shut down. And then there's this really cool effort that I can't take credit for, the Move Bond initiative. This is led by some of my colleagues at the Smithsonian, as well as a handful of other institutions. For those that don't know, biodiversity observation networks are sort of vehicle data move into these big global multinational policy frameworks. So Convention on Biological Diversity and Convention for the Protection of Migratory Species. So if you heard about like the COP and the Kunming-Montreal post-2020 biodiversity framework, all the data that drives these indicators comes from a group called GeoBond. And GeoBond has thematic bonds that nest under it that deal with particular realms. There's like one for marine and then different data types. We are proud to announce that in the last month, ours was approved. So we're now the sixth thematic bond, focused on movement data alone. We have a paper in review sort of highlighting why and what movement data can do for international policy frameworks. Like what can we help them see that they're not? Because right now it's all occurrence data, right? I'm leading a couple of the working groups under this, one to develop a mortality indicator, and then another to help us actually find data, right? We need to be able to discover it in order to use it, and it turns out to be a key problem. But if you're interested in this, if you're somebody that's interested in policy in particular, there are going to be continued public announcements and ways to get involved. This is really starting to take off, like right now. And so, with that, I will stop and take questions. I will say my thank yous. There's a tremendous amount of work and collaborators that go into all of this, and especially the folks that share their data. I think it's important. It's really important to highlight how much work goes into collecting the data from just one animal. And so, you know, I was showing you bar charts of, you know, like 4 percentages. Underneath that were thousands and thousands of hours of people putting, you know, finding animals, putting tags on them, processing the data, and then choosing to share them with us to be able to do that. And I'm incredibly grateful for all that. It's important to stop and acknowledge those folks. And with that, I'd be happy to take some questions.
Thank you for that wonderful talk. Something that I'm sure you always think about is how are the tags themselves like affecting behavior. So I wondered particularly with that migratory survival graph, like do you think that's a overestimate of the actual mortality that would occur if the bird was not tagged? Or
it's the thing that nobody likes to talk about, right? And it's very hard to measure.
Yeah.
Because if we need the tag to know if the animals, what the survival rate was,
then, right? Control. Yeah.
It's like the World War II plane problem, right? There are a couple places where people studied it. We never published it. We did it with apples because we have long-term banding study there, and so we can sort of like use the breeding adults to say like who have we tagged. There's so, you know, 40-year data set, and it reduces survival. You know, the biggest question for me is whether that survival interacts with your covariate of interest. And it's hard to say. It is totally could have, right? If we think like the mechanism of the reason we go after the drying is because we know that low EVI, drier Caribbean leads to lower insect biomass, even birds are in poorer conditions. So Pete Mara has a whole Red Star system where they looked at this in a bunch of like very discrete mechanistic studies that are really cool. And so you could imagine that carrying a tag would interact with that. So certainly something we can't totally rule out. That said, one of the reasons those automated telemetry tags are super cool is that they're really, really small. So we're not right at the threshold of what we're allowed to do.
Okay.
Yeah, there is this push in movement ecology. Sorry, it's a long but push in movement ecology to like put the coolest tag you can on your animal, like get right up to that 3 or 5% weight limit. And I think for the demographic, we need to think a lot harder about that. We should actually be trying to like. I go to these movement ecology conferences and I was like, can you make me a tag that records once a week? And like, I don't need the best specialist. I want to not have to catch my animal and go for a really small tag. Yeah.
I was wondering, does all these initiatives, so this is obviously very macro ecology, do they have any initiative to expand it to like a microbial ecology and dispersal? Because I think the concepts are similar. We draw a lot of differences from macro.
Do you mean like microbes themselves?
Like the microbiomes and everything, like how their migration is affecting all those microecology.
Yeah, I would love to do that. So we're putting together a proposal right now around this ecology of death idea, and we do work in a building within the microbial ecology. They think about this from an endangered species standpoint. So they're like involved in the black-footed parrot releases. And so for them, you know, every animal is really expensive and really valuable. Not just valuable for money, but like it's, you know, the species continued existence is based on it. And so they do a ton of work trying to understand, can you predict the outcome for those individuals based on their microbiome? And so we're trying to get them integrated in this project to do that with wild animals. So yeah, does your gut microbiome interact with these environmental experiences to modulate your mortality outcomes? Which I think would be really cool. The other way around, where now you're trying to move, like, how does the migration move the microbes around? The disease ecologists are thinking about that, but outside of disease ecology, I don't think. There's a lot of disease ecologists thinking really hard about movement ecology right now. So there's sort of like obvious potential there. There's also big problems, like just big methodological problems to solve, but there's a ton of problems. So yeah.
Thank you.
Yep. Yeah.
It's got awesome talk.
Thanks.
I was curious about two components. One, you're talking about the life history trade-offs, and then the ecology of death. GPS tracking devices to identify mortality events and the environmental conditions that preceded those. But it seems like the other side of that equation is birth, right?
Yeah.
Yep. And so at least in my little world that I tend to work in with large mammals, ungulates, we use these GPS collars to not that many people actually do behavioral stuff. It's a lot driven by demography of big game, hunted, economically important species. And so we have things like badger implant transmitters that communicate with the collars to tell you that they gave birth. And you go out and see how many offspring were produced, tag them, track their survival to get custody, recruitment, and adult survival. And that's really key for these demographic models with the species I work with. That's obviously not possible at scale, right? These like huge, you know, maybe. Like I guess I was just thinking like there have been some efforts to use the behavioral data to try to identify birth events.
Right.
And if you could maybe also integrate an ecology of birth, then it wouldn't be so common.
Yeah, it would. Better messaging. Totally. Yeah, 100%. It is the obvious flaw in this. And I think, I mean, like you've done work showing the importance of considering fecundity in ungulates in particular. So it's something we need to get better at. Absolutely. I think there's a couple things to say. I mean, one is that for most species, it's the mortalities that drive the population declines. So from a conservation perspective, it is rarely, but not never, it is rarely just like a fecundity gap that is driving this time and wellness that we know. What that is. But yeah, from a life history perspective, you need both. And it would be great to understand how that modulates. And ungulates are far ahead because you have these vaginal implant tags. There are people that are trying to do it from the behavior, right? Like Corey Overton, UPF had a paper where just sort of like, did you behavior that looks like sitting on a nest? Did you sit on a nest for the number of days that you would expect you to sit on a nest for your eggs to like close to fecundity? I think that there's other methods out there if we start getting really creative. I mean, I have this like harebrained idea that I am embarrassed to tell a funder about. I mean, you could put microphones on a tag, right? I mean, birds make noise. And I know the number of days that fledglings should be making noise in a nest. I mean, people put microphones on tags. That's not unheard of. Like we could train AIs to process those data or train a simple in Markov models to process those data and ask, like, did you get to a behavioral state where you even separate how many chicks there were? So that's what I mean by, like, when I say we need new technology, like, it's great to toot our own horn and show like we've gone from accidental survived hunting attempts to radio taxonomy, but gosh, there's still a ton more that we need to do, and that's ultimately where we'd like to do. Part of the reason we're pushing on mortality is like it's close because we know we can do that for a lot of species right now. And I think like with automated telemetry, we're a couple of methods away from being able to do that with dozens of species. And we don't need to use technology, we don't need powers, we just need to figure out the movement models for that particular detection system. So that's a lower hanging fruit. It just is the answer.
Yeah. Building on that with another vital rate that I mentioned there, a lot of my background was how immigration gets incorporated into plotting trajectories for different populations. And it seems like it'd be really within the realm of, maybe this is already being done as well, but sort of defining different subpopulations or metapopulations with tracking these individuals and seeing that will reflect variation and movement between the sites.
Yeah, you can. Doing this, right? And, it makes it harder than you think.
You need really comprehensive, right? And so, like, when you
Get into any of these things that
start to approach something like a network analysis, missing this is just really awful, right? Like,
missing at random in a network
still produces bias in your network measures. And so when you're talking
about population connectivity,
or, I think about
migratory connectivity,
missing this is still a problem.
but within
Systems that are constrained or really comprehensive tagging efforts,
there are certain species that we're pretty good at catching, and we have robust programs. So like there are groups that are talking really ambitious. They're like Martin, he's talking about tagging every white stork in Germany. It's insane. It's an insane thing to say, but he's like, I don't know.
they put an antenna on the space station.
They're not the buildings.
There's people that, they know where
every nest is. They do,
maybe that's not crazy.
Maybe it's crazy, but so yeah, I mean, I think, yeah, you can do that. The other thing that's exciting that's related is connectivity modeling and validating connectivity modeling. So again, that's the other indicator, and that indicator space that we're really pushing is right now that's done with the habitat. So that's done with simulation model, resistance model, where we think the connectivity should be, is almost never confronted with data. Now we have the data and actually ask, does this quarter matter or not? I think that's pretty.
Okay. Cool. Thank you so much.